Are you able to see the screen? Are you able to see? Are you able to see or not? If if you will not able to see the next slides, you please tell me. Live chat I have. I'm observing continuously live chat also. Okay. So if you will not able to see the screen, now I am going to explain. If you will not able to see the screen, then you please inform me. Okay. And next slide also. Now the slide which is displayed here is the slide of transamination. Transamination is now display. I have displayed the transamination slide, but when I will teach the next to transamination, if you will not able to see that slide, you please and please inform me, so that I will be able to solve this problem. This is not problem from my side, this is the problem to YouTube. I have inquired to other staff members also. This is the problem to YouTube and that StreamYard. So next slide you may not able to see in the last lecture, but now you will be able to see it. So I am going to teach now transamination. This is the trial whether you are clearly able to see or not. So this is the trial, but I will teach transamination in this lecture. So shall I start? Yes or no? So what is transamination? The transfer of amino group NH2 group from the amino acid to keto acid is known as transamination. Transamination is the metabolic pathway for the degradation of amino acid. All the amino acids are catabolized or degraded by the process of transamination and deamination. The transamination is the transfer of amino group from the amino acid to keto acid known as transamination. You have already studied this in the DYBSc level. First, I am covering all those topics which you have already studied it, so that it will be easy for you to prepare notes and to do the study of all these topics. Then we will take all these topics which you have not already studied at the SYTY level. There are only one topic, that is the membrane transport, which you have not studied. Maybe the carbohydrate, amino acid, protein, nucleic acid you have already studied in the FYBSc or SYBSc. So it is not the point. So I'm going to deal with now transamination, that is the transfer of amino group from the amino acid to keto acid is known as transamination. This process involves the interconversion of pair of amino acid and a pair of keto acid catalyzed by a group of enzyme called transaminases or aminotransferases. Process involves the interconversion of pair of amino acid, that is the two amino acid take part in the transamination reaction. Two amino acid take part and these two amino acids are interconverted to form the keto acid. Means one amino acid is converted to keto acid and another keto acid which acts as a reactant, that keto acid is converted to amino acid. And this reaction is catalyzed by the enzyme transaminases or aminotransferases. So what are the important features of the transamination? All transamination requires pyridoxal phosphate. Keep in your mind, pyridoxal phosphate, a coenzyme derived from the vitamin B6. All transaminases require pyridoxal phosphate, a coenzyme of vitamin B6 for the transamination reaction. This may come to the multiple choice question, that which amino acid is involved in transamination reaction, or the number of coenzymes will be given, and which of the following is involved in the transamination reaction. It may come to the exam. So all transaminases require pyridoxal phosphate, a coenzyme derived from vitamin B6 for the transamination reaction. Specific transaminases exist for each pair of amino and keto acid. Specific transaminases exist means for the alanine, it is the alanine transaminase. For the glycine, it is the glycine transaminase. For the isoleucine, or for other any enzyme, other any amino acid aspartate, it is the aspartate transaminase or alanine transaminase. That is, for each amino acid there is a specific transaminase enzyme which take part in the transamination reaction, which makes significant contribution for transamination. There is no free ammonia liberated during the transamination, only the transfer of amino group occurs. There is no free ammonia liberated, only the transfer of amino group occur from amino acid to keto acid. So keto acid is converted to amino acid and amino acid is converted to keto acid. So transamination is reversible reaction, not irreversible, it is the reversible reaction. Amino acid, there is a specific transaminase enzyme which take part in the transamination reaction. transamination is contribution for transamination. There is no free ammonia liberated during the So I have checked that on YouTube also the second slide is displayed. Is it? I am also checking simultaneously on my YouTube. So uh, second slide, that is the point number 3. That is, there is no free ammonia liberated. Only the transfer of amino group occurs. This is displayed now properly. And for the ED pathway, for the fatty acid degradation, and for the fatty acid synthesis also, I have displayed all slides. But because of the problem to this YouTube, you was not able to see it. It is not problem from this side, because some of the staff member, they also have same problem. So again I will repeat, there is specific transaminase for the specific amino acid, like aspartate transaminase for the transamination of aspartate residue, then alanine transaminase to catalyze the reaction of alanine transamination. Like this way, there is no free ammonia liberated in the reaction, only the transfer of amino group takes place. Then transamination is a reversible reaction. Transamination is very important for redistribution of amino group and the production of non-essential amino acid. Very important for the redistribution of amino group. Redistribution of amino group means amino group from the amino acid is transferred to keto acid. Keto acid accept that amino group and it is converted to amino acid, and whatever the keto group is releases from that keto acid is taken by that amino acid from which amino group is removed, and that amino acid group when removed it accept the keto group and it is converted to keto acid. So this reaction is the reaction of amino acid 1 plus keto acid 1 converted to amino acid second plus keto acid second. Or it may be the amino acid 1, keto acid second converted to amino acid second and keto acid first. Like this way, two different forms are given in the book. So this is interconversion of amino group. Only transamination is the interconversion of amino group. So this is very important for redistribution of amino group and the production of non-essential amino acid for the requirement of the cell. It involves both catabolism as well as the anabolism. That is, it also involves the synthesis process. And catabolism, that is the degradation process. Both are involved in the transamination. Transamination diverts the excess amino acid towards the energy generation. It diverts excess amino acid towards the energy generation. And trans amino acid undergoes transamination to fully concentrate nitrogen in the glutamate. Amino acid undergoes transamination process to finally concentrate nitrogen in the glutamate. Glutamate is the only amino acid that undergoes oxidative deamination. It undergoes oxidative deamination process to liberate free ammonia for the urea synthesis. All amino acid except lysine, threonine, proline and hydroxyproline participate in transamination. All the amino acid, that is, there are 20 amino acids. So out of the 20 amino acid lysine, threonine, proline and hydroxyproline, they participate in transamination reaction. You should keep in mind all these 4 amino acid lysine, threonine, proline and hydroxyproline. That is the amino acid which does not participate in the transamination reaction. Transamination is not restricted to alpha amino group. It is not restricted to alpha amino acid only, that is the alpha amino groups. Delta amino group are also transaminated. That is the amino acid having the alpha, beta, gamma, delta amino group. They also undergo the transamination reaction. Then serum transaminases are important for diagnostic and prognostic purposes. That is, serum transaminases are these transaminations. Doctors recommend you analyze or you check your serum transamination, that is to maintain the level of HGPT, HGOT level in the blood. So these are the serum transaminases. These are they have the diagnostic significance, and they are related to the diagnosis, that is level of that serum transaminases HGPT, HGOT make decreases in the blood or may increases in the blood. Okay, so these are the important features of transamination. Then what is mechanism of transamination? So transamination occurs in two stages: transfer of amino group to the coenzyme pyridoxal phosphate bound to the coenzyme to form pyridoxamine phosphate. The amino group of pyridoxamine phosphate is then transferred to keto acid to produce new amino acid, and the enzyme PLP, that is the pyridoxal phosphate transaminase. This enzyme is regenerated. All the transaminases require pyridoxal phosphate as a derivative of vitamin B6. Okay. So what are the mechanism of transamination? The transamination occurs in two stages: transfer of amino group to enzyme pyridoxal phosphate, coenzyme pyridoxal phosphate to form pyridoxamine phosphate. Pyridoxamine phosphate then transfer amino group to keto acid to form new amino acid, and amino acid with the pyridoxal phosphate, it is regenerated. So all transaminases required pyridoxal phosphate, a derivative of vitamin B6. So this is reaction. Are you able to see this reaction? This is reaction. This is amino acid 1, R1, CH, C double bond O, NH3. This NH3 is the amino group of amino acid first. Okay. This amino acid first, it reacts with the keto acid second, R2, C double bond O minus COOH. Okay. The COO minus are the COOH. COOH, that is the carboxylic group. R1 is the long chain, whatever the chain present in that amino acid. So R1CH NH3 amino group and COOH, this first amino acid, plus R2CO, that is the keto acid, COOH, this keto acid, 1 plus 2 with the pyridoxal phosphate PLP with the help of enzyme transaminase it is converted to keto acid first, that is R1. See here, R1 is as it is. This NH3 group is replaced by O group, that is amino group is replaced by keto group, and amino acid is converted to keto acid, R1CO COOH. This is the conversion of amino acid to keto acid, and this keto acid second, that is the R2CO O minus COOH. From this group, O minus get releases and it accept amino group and it is converted to R2C NH3 means CHNH3 COO minus, that is the amino acid second. So what is this reaction? Amino acid first plus keto acid second with the help of pyridoxal phosphate as the coenzyme with transaminase enzyme is converted to keto acid first. Amino acid first is converted to keto acid first, and keto acid second is converted to amino acid second. So this is only the interconversion of amino group and keto group takes place. So this is catabolic as well as the anabolic process. Amino acid second is regenerated, that is the anabolic process, and this amino acid first is degraded. This amino acid first is degraded and converted to keto acid first, and keto acid second is again degraded and it is converted to amino acid second. So this is catabolic as well as the anabolic process. And how this catabolic and anabolic process? It is only interconversion of amino group from amino acid first to keto acid second. Keto acid second is become amino acid second, and amino acid first, from which amino group is releases, it accept keto group and it is converted to keto acid first. So this is the mechanism of transamination, which is carried out by the enzyme transaminase. This is example. This RCHNH2 COOH plus keto acid RCO COOH, same reaction with glutamate transaminase, enzyme glutamate transaminase, and required pyridoxal phosphate. So it is converted to glutamic acid. Amino acid is the form is the glutamic acid, and keto acid form is the alpha ketoglutarate acid. Means this amino acid is converted to alpha ketoglutarate acid, and this keto acid is converted to glutamic acid with the help of enzyme glutamate transaminase. This is again one more example of transamination. This is the alanine CH3CHNH3 plus COO minus alanine amino acid alanine plus alpha ketoglutarate CO COOH, CH2, CH2, COOH, that is the alpha ketoglutarate. Alanine plus alpha ketoglutarate undergoes reaction by enzyme alanine aminotransferase. Alanine aminotransferase transfer amino group from alanine the alpha ketoglutarate, it is converted to pyruvate. Means from where pyruvate is generated, this alanine CH3 C double bond O COO minus, this alanine is converted to pyruvate and alpha ketoglutarate is converted to glutamate. This is the best example of alanine aminotransferase reaction and example of transamination reaction catalyzed by enzyme alanine aminotransferase. I will again explain. Alanine CH3CH NH3 plus COO minus. This alanine is converted when NH3 group is removed. This alanine is converted to pyruvate CH3CO COO minus, and alpha ketoglutarate when reacts with the alanine. O group is removed from the alpha ketoglutarate and is converted to glutamate, that is COO minus CH NH3 plus CH2 COO minus, that is the alanine catalyzed by enzyme alanine aminotransferase, and the product form is the glutamate. Okay. So have you any difficulty in transamination? I think I have finished transamination. That is the transfer of amino group from the amino acid to keto acid. What are the reactant of the reaction? Reactants are one amino acid plus one keto acid with the PLP as a coenzyme, and amino transferase is enzyme which is converted to amino acid is converted to keto acid, and keto acid is converted to amino acid. Do you able to see all the slides? Have you seen all the slides until the example of transamination? This is the last slide, and I have shown all the slide. And uh, last week it was the problem with this StreamYard and YouTube. That's why you was not able to see all the slides. Because I have seen on the YouTube, C3 pathway, C4 pathway is displayed properly. That is the Microsoft Word document, but C4 pathway was not displayed. What is your difficulty? How does NH3 add in the reaction of pyruvate to glutamate? So it is added NH3. NH3 is removed from the alanine. Okay, it get releases from the alanine by the enzyme transaminase. It is accepted by the PLP, that is the pyridoxal phosphate. Pyridoxamine phosphate is formed. That pyridoxamine phosphate then donates that amino group to keto acid, that is the alpha ketoglutarate. The alpha ketoglutarate is converted to glutamate. When alpha ketoglutarate will accept that amino group, it will convert it to glutamate, and alanine, from where the amino group is released, that amino group when released, that fill in the blank is that filled by the NH3. Whatever the space created, that space is filled by the NH3. Then alanine is converted to pyruvate. Means NH3 when removed, it accepts O, oxygen, keto group. It accepts keto group and it is converted to pyruvate, and alpha ketoglutarate which accept NH3 group, it is converted to amino acid glutamate. Clear, Sam? Do you get? See, only four amino acid: lysine, threonine, proline, and hydroxyproline. Okay, keep in your mind proline and hydroxyproline. Same, and lysine and threonine. Only this four amino acid does not take part in the transamination reaction. All other amino acid, alanine, glycine, histidine, then isoleucine, then valine, tryptophan, tyrosine, all the amino acid take part in the transamination reaction. So what happens? Amino group from that amino acid is releases. That amino group is accepted by pyridoxal phosphate. Pyridoxal phosphate accept that amino group, converted to pyridoxamine phosphate. Pyridoxamine phosphate. See here, it is clearly mentioned. Pyridoxamine phosphate donates that amino group to keto acid. So the keto acid is converted to amino acid, that is suppose here glutamic acid, and amino acid is converted to alpha ketoglutarate acid, that is the keto acid. So this is the reaction takes place in the transamination reaction. Okay, what difficulty you have? Now I am going to show you deamination. We are not going to break this lecture. Instead, we will see deamination right now. Okay. We will see now deamination. You don't leave. Immediately I will show you deamination. Are you able to see the screen of deamination? Are you able to see the screen of deamination? I have now display the screen of deamination. Are you able to see it? Before that, what is the difficulty of SAM? Why does they do not participate in transamination? A single reason: do they possess same chemical nature? Like that. Why this amino acid does not participate in transamination? Because there may not be the existence of specific transaminase enzyme for that amino acid. There is specific transaminase enzyme exists for each amino acid. So that amino acid which have transaminase enzyme, they participate in transamination reaction. Those amino acid which do not have the transaminase enzyme, they do not participate in transamination reaction. So there may be that cell do not have the enzyme for that specific transaminases. So that for transaminases does not participate in transamination reaction. Okay. Now I'm going to see you deamination. Are you able to see deamination? What is deamination? The removal of amino group from the amino acid as NH3 is known as deamination. Removal of amino group from the amino acid. Here is the completely removal of amino group takes place in the deamination. While in the transamination only transfer takes place, in the deamination only amino group is removed is known as deamination. So deamination results in the liberation of ammonia. for urea synthesis. Deamination results in the liberation of ammonia. It results into the liberation of ammonia NH3 for the urea synthesis. Simultaneously, the carbon skeleton of amino acid is converted to keto acid. That is, amino acid is converted to keto acid with the complete removal of ammonia NH3 group from the amino acid in the form of ammonia. So deamination may be either oxidative or non-oxidative. So what is oxidative deamination? Oxidative deamination is the liberation of free ammonia from the amino group of amino acid coupled with the oxidation. Oxidative deamination is the liberation of free ammonia from the amino group of amino acid coupled with the oxidation. Oxidation reduction reaction takes place in the oxidative deamination, hence known as oxidative deamination. Liberation of ammonia takes place with the removal of amino group with oxidation reaction. So this takes place mostly in the liver and kidney. The purpose of oxidative deamination is to provide ammonia for urea synthesis. The purpose of oxidative deamination is to provide ammonia. NH3 is provided for the synthesis of urea. Urea get excreted through the urine. So the purpose of this is applicable to the animal cell or human cell where this oxidative deamination takes place. It takes place in the liver and kidney, in which NH3 is completely removed from the amino acid. Now we are taking pulses, we are eating pulses, and most of the other protein containing diet. So that protein containing diet, it is made up of amino acid, and that amino acid, when it undergoes degradation in the body, from that amino acid, when it undergoes by the oxidative deamination process, oxidative deamination process, it removes ammonia. It completely removes ammonia, the NH3 from the amino acid for the urea synthesis. So this takes place in the liver and kidney. And it generates alpha keto acid for the variety of intermediate formation of an sorry. The purpose of oxidative deamination is to provide ammonia for the urea synthesis and alpha keto acid for the variety of intermediate formation. And this takes place with the intermediate form is the alpha aminoglutarate. And this reaction is catalyzed by enzyme glutamate dehydrogenase, which reversibly link to the glutamate metabolism with the TCA cycle. Okay. So glutamate dehydrogenase is involved in both catabolic as well as anabolic reaction. Are you getting me? Here only what I want to tell you is the purpose of oxidative deamination is to provide ammonia for the urea synthesis. Okay. This is my first sentence. Then alpha keto acids are formed, which are utilized for other which are acts as a intermediate for other metabolic reaction. And this takes place with the formation of one more intermediate reaction takes place with the formation of one more intermediate, that is the alpha aminoglutarate. I will show you reaction. And reaction is catalyzed by enzyme glutamate dehydrogenase. You first see this reaction, glutamate by the enzyme glutamate dehydrogenase. This reaction required NADP, which is converted to NADPH, and glutamate is converted to alpha aminoglutarate. This is the intermediate C double bond NH2. This is the intermediate. Alpha aminoglutarate is then converted to alpha ketoglutarate. by enzyme glutamate dehydrogenase. Both the steps are carried out by only one enzyme, that is glutamate dehydrogenase. And complete removal of NH3 in the form of ammonia, NH4, takes place. NH3 plus one more hydrogen, that is NH4, is removed in the reaction. Reaction is catalyzed by enzyme glutamate dehydrogenase, and glutamate is converted to alpha ketoglutarate. C double bond O, CH2, CH2, C double bond O, that is the keto group, and COO. That is this COO, these are the carboxylic acid group, COOH, COOH. And COOH, CH2, CH2, C double bond O, that is the keto group, and COOH, that is the complete structure formation is the alpha ketoglutarate. And both reactions are catalyzed by enzyme glutamate dehydrogenase. Today what happened, I am going to check simultaneously on the YouTube whether all the slides are properly displaying or not, because actually we do not have to check, but since from last 2 to 3 days, because all the slides were not displayed, so today I am taking care of whether all the slides are properly displaying on the YouTube or not. Otherwise only first slide you are able to see, and other slides I am going to uh, teach only. I used to talk, and I don't know whether you before 2 to 3 days, I don't know whether you was able to properly see or understood or not. Okay, so what is the role of glutamate dehydrogenase in the process of transamination? The amino group of the most of the amino acid is transferred to alpha ketoglutarate, and that alpha ketoglutarate is produced to glutamate. Glutamate acts as a primary collection center for the amino group. What happened? Pay your attention again here. In the process of transamination, the amino group of the most of the amino acid transferred to alpha ketoglutarate, and alpha ketoglutarate and other amino acid form is the glutamate amino acid. Okay, that glutamate which is now formed in large amount in the body, that glutamate have to undergo again degradation reaction, and it undergoes degradation reaction by the enzyme, by the process oxidative deamination, and by the enzyme glutamate dehydrogenase. Are you getting me? Whatever glutamate, a large quantity of glutamate is accumulated in the body, that glutamate is undergo degradation by the oxidative deamination process with the help of enzyme glutamate dehydrogenase to liberate free ammonia. Ammonia is liberated. This enzyme is unique that it utilizes NAD plus or NADP plus. Either you write down NAD plus, it is converted to NADH and H plus, or NADP plus, which is converted to NADPH or NaH plus. It can utilize either NAD plus or it can utilize either NADP plus as a coenzyme to convert that amino acid to alpha immunoglutarate. Alpha immunoglutarate is converted to alpha ketoglutarate. So conversion of glutamate amino acid to alpha ketoglutarate, it takes place with the formation of the intermediate alpha immunoglutarate with the help of enzyme glutamate dehydrogenase, and NAD plus or NADP plus acts as a coenzyme for the reaction. Are you getting me or not? Sometime you may not able to see the screen clearly, because it may be due to the range problem. From my side I am showing you, but it may be because of the range problem. Okay, so what is oxidative deamination? I have explained oxidative deamination. I will again explain what is oxidative deamination. I have explained what is the role of enzyme glutamate dehydrogenase. Glutamate dehydrogenase is a unique enzyme which required either NAD plus or NADP plus as a coenzyme, which can utilize NAD plus or either NADP plus. Whatever available in the cell, it is able to utilize that coenzyme. Now, how this glutamate dehydrogenase is unique in the process of transamination we have just seen before the 10 minutes. So what happen in transamination, which whatever amino acid is involved, example alanine aminotransferase, amino acid alanine or amino acid glycine or amino acid valine, whatever the amino acid is involved in the reaction, all the amino acids are converted mostly to the glutamate. All that amino acids, it undergoes catabolism and these are converted to amino acid glutamate, and then glutamate is concentrated into the body. The large amount of glutamate is produced into the body, so that glutamate again have to undergo the degradation, otherwise the glutamate concentration will takes place more in the body, its concentration will increase. So glutamate have to again undergo the degradation by the oxidative deamination process. So it break down by the process of oxidative deamination. And what happened during the process of oxidative deamination? Degradation of glutamate takes place by the complete removal of amino group NH3 group in the form of ammonia NH4, and the reaction takes place in the two steps. uh, Amino acid glutamate is converted to intermediate alpha aminoglutarate, and alpha aminoglutarate is converted to alpha ketoglutarate. And for this reaction, NADP plus or NAD plus is acts as a coenzyme, and reaction is catalyzed by enzyme glutamate dehydrogenase. So this reaction liberates ammonia. Free ammonia is liberated. I have again show you this slide. See here, free ammonia is liberated, and that ammonia is excreted from the body in the through the urine. Through the urine, this ammonia it get excreted through the body. Why urine have the smell of ammonia? Because large number of whatever the ammonia present in the body, that ammonia get excreted through the urine. Okay, so do you get again, or you have any difficulty? So now I am going to teach you next oxidative deamination by other enzyme. Oxidative deamination, whatever now I explain, is the oxidative deamination by the amino acid, glutamate dehydrogenase. Now I am going to show you oxidative deamination by amino acid oxidases. So this is oxidative deamination by amino acid oxidases. So L amino acid oxidase and D amino acid oxidase, these are the pleoprotein possessing FMN and FAD as a coenzyme group respectively. So they acts on the corresponding amino acid, L amino acid or D amino acid, to produce their corresponding keto acid and liberates NH3. So in this reaction also oxygen is reduced to H2O2, which is later decomposed by the enzyme catalyst. L amino acid oxidase and D amino acid oxidase are the fleoproteins, and they carry out oxidative deamination process. We have seen the oxidative deamination process by enzyme glutamate dehydrogenase. Now we are going to see the oxidative deamination process by enzyme L amino acid oxidase. See here, enzyme L amino acid oxidase and enzyme D amino acid oxidases. So oxygen is reduced Oxygen reduce to H2O2, which is again decomposed by the enzyme catalase. We will see that reaction. So D-amino acid mostly found in the plant and microorganism, they which are not present in the human being or in the mammals. D-amino acids are not present in the human being. They are mostly present in the microorganism. So that D-amino acids, these are metabolites by the amino acid oxidases and converted into their keto acids. And alpha keto acid, which is produced, it undergoes transamination to be converted into the L amino acid, which participate in various metabolic reaction. So keto acid may be oxidized to generate energy and serve as the precursor for glucose and fatty acid synthesis. So D amino acid oxidase, this is the important because it converts D amino acid to L amino acid. Are you getting me? D-amino acid oxidase, this is the important enzyme because it converts D-amino acids to L-amino acid in the body. L, human being have mostly L-amino acid. So when L-amino acid it undergoes degradation, it produces ammonia. But when we take D-amino acid, sometimes they also undergo degradation. But body is not able to synthesize D-amino acid. So these are found mostly in the plants and microorganisms. With the help of enzyme D-amino acid oxidases. Okay, so oxidative deamination by amino acid oxidases, that is, it involves the role of L-amino acid oxidase enzyme and D-amino acid oxidase enzyme. Okay, now next is non-oxidative deamination. Till up to this, we have seen oxidative deamination. Now, what is non-oxidative deamination? Only one slide is remaining from the deamination. So, I am going to finish it. So, non-oxidative deamination, some of the amino acid, they can be deaminated to liberate ammonia without undergoing oxidation reaction. That is, the oxidation reduction reaction cannot take place. So some of the amino acids, they are deaminated to liberate ammonia without undergoing oxidation. So amino acid dehydrases, these are the enzyme amino acid dehydrases, which catalyzes the conversion of serine, threonine and homoserine into their hydroxy amino acid, and they undergo non-oxidative deamination process by the PLP dependent dehydrases enzyme dehydrases or dehydratases serine, threonine, homoserine. With the help of enzyme dehydratases, they undergoes dehydration reaction or non-oxidative deamination reaction to convert into the respective keto acid, and ammonia NH3 is liberated. Then amino acid desulfahydrolase, this amino acid desulfahydrolase, they again catalyzes the conversion of sulfur containing amino acid, namely cysteine and homocysteine, to undergo the process of deamination with the desulfahydration to produce keto acid. See here, cysteine or homocysteine by the enzyme desulfahydrolase is converted to pyruvate plus NH3 plus H2S. So cysteine and homocysteine, they undergoes deamination by the process of desulfahydrolase enzyme to produce pyruvate, NH3 and H2S. Then deamination of histidine. Amino acid histidine undergoes again deamination process by enzyme histidase to liberate ammonia and urocanate. Urocanate is one of the end product. Again, I will repeat. Non-oxidative deamination, actually transamination, oxidative deamination, and non-oxidative deamination you have already studied in the TYBSc level, and you have given the exam of TYBSc just in the December by studying all that material. So, and I think from April to December again you have studied all the material. You have given the exam of enzymology, but some of the students who have not passed the metabolism paper, they might have studied the course of metabolism also in the in the last sem, that is in the sixth sem. But you are all the topper candidate who have taken the admission for the MSc, so your paper was clear in the fifth sem only. But actually one you have studied it one time, so this is easy for you. You once you go through the notes of TYBSc, the students of the Shahada College, I have given you already in the notes of TYBSc. You one time you read that notes of transamination, oxidative deamination, and non-oxidative deamination. Now non-oxidative deamination, again I will repeat, it takes place that it is the complete removal of amino group with the liberation of ammonia without undergoing oxidation reduction reaction. Here there is no involvement of NAD or NADP plus. So there is no oxidation reduction reaction. Only removal of amino group takes place. That is the deamination takes place. Okay, then amino acid, this non-oxidative deamination takes place by the enzymes amino acid dehydratases, amino acid desulfahydratase, and amino acid histidine. It undergoes deamination by the enzyme histidase. Okay, so amino acid dehydratases catalyzes the reaction of serine, threonine, and homoserine deamination to conversion into their respective keto acid. Amino acid desulfahydratase catalyzes the conversion of sulfur amino acid, that is cysteine and homocysteine, into their respective keto acid, that is the pyruvate. Here also pyruvate is generated. In the first reaction, pyruvate is also generated in this reaction. Here also pyruvate is generated. And here in the histidine degradation, histidine deamination, urocanate is liberated with the ammonia as the important product. NH3 is liberated during the reaction. So this with this I have finished transamination, oxidative deamination, and non-oxidative deamination. Do you have any difficulty? Purine and pyrimidine synthesis, one more point which already you have studied. Now I am not going to take it. I will take it lastly if time will available. Now I will take tomorrow I will take membrane transport, because membrane transport you have not studied it before. Okay, do you have any difficulty? No difficulty. So shall I stop this lecture now? 1 hour. Since from last 1 hour you are means you are listening since from last 1 hour. Maybe become bore. But actually today I want to take lecture, and I have to see whether properly all the slides you are able to see or not. So all the slides I have also check on YouTube simultaneously. I am also observing lecture on YouTube that all slides are properly displayed now today. So you will be able to prepare your notes from that slides, and you might have all these notes in the TYBSc level. So shall I end now? Uh, One more point from the next lecture, I will not see the YouTube, because when I am simultaneously observing YouTube and I am teaching you, my some of the concentration goes to the YouTube lecture and some of the concentration remains here on the laptop. So you go through, you see that lecture, and if any problem, you interrupt me, send the message that all the slides are not displaying properly, so that I will be able to discontinue, and again I will able to share the link of the new lecture. Okay.